In the following 15 minutes, I would like to present the forefront of analytical developments with focus to reveal what we are calling emerging contaminants hazards, which belong to the chemical universe we are living in. Every day we are using hundreds of thousands of chemicals that through several point sources are released into the environment, either air, water, marine environment, whatever. And we have in the list few hundreds of substances that are regulated, but the rest of the iceberg is hidden, mostly unknown, and with unknown properties, toxicological or fate-related properties. Therefore, we need methodologies to reveal this, investigate, and reveal the hidden pollution, contamination, and if we take into account that these chemicals are released from several point sources and are in contact with biotic processes, we have a range of potential metabolites or transformation products that make the problem even more complex. To do so, to investigate this hidden pollution, we have developed a group dedicated to this investigation with focus. The core group is developing methods, and we apply these methods to environment, life sciences, and food, of course. And the most analytical techniques that we are using are related to mass spectrometry. So we have a holistic research approach. We try to develop the methods and apply it to the overall environmental compartments, not only the aqueous phase, but also soil, sediments, air, and living organisms, producing data, which are most of the time big data, and then through our collaborators, we are participating in a network called Norman. We are in the core group there, but we have links to ECA, EFSA, EU, EPA, and DG Environment. We are trying to, let's say, push forward the change of chemical management in Europe and beyond. Not only priority pollutants and known emerging pollutants are sought for, but we develop methods to explore the whole, let's say, diversity of chemicals in our samples without prior knowledge. This is a very difficult task because you know that since we are aimed to use these methodologies for regulatory purposes, we should define the scope of the methods. Therefore, how we define the scope of non-targeted analytical methods, this is another aim of our research and effort. In any case, we define the scope with several complementary orthogonal techniques like hydrophilic liquid chromatography, GC, and reverse phase chromatography. We apply these methodologies to a range of samples from our campaigns, and the data produced are processed through 3 distinct workflows. Target analysis means that we have the standards in the laboratory. We have more than 3,500 standards for several different analytical techniques, validated methods, pure quantitative results. We have the suspect screening. This was something that we have the wide scope target and wide scope suspect screening. It's an initiative for more than 10 years that I'm trying to apply in several campaigns. Suspect screening means that we develop the libraries with suspected substances, with all the mostly in silico properties, identification points, as we say, in order to treat the data, process the data, and have tentative identification of hazardous substances, and then not only we stay at this point, but we have developed algorithms that producing semi-quantitative results without the need of standards, alleviating the need of standards, meaning that we have, even with high uncertainty, quantitative results from this screening process. We expand the data set with other more substances with quantitative. Why we need quantitative results? Because we need this for risk assessment. Without quantitation, you don't have, the means to do risk assessment. So we are using a range of mass spectrometers. These are spectrometers that have also additional capabilities for higher rate of identification. We use also this TIMS dimension, means that we have ion mobility data for a fourth dimension of identification. And of course, these instruments are very expensive, but also very powerful in identification. This is the databases that we have for the targeted analysis, both GC and LC amenable substances. And for 1,500 compounds now, we have also the TIMS dimension, the CCS values, meaning that we have increased rate of identification. And apart from the software that we are using from Bruker, for instance, for targeted analysis, we have developed now, let's call it infrastructure, which means that this digital sample freezing platform, DSFP, was developed to screen, to apply this wide scope suspect screening. Has all the algorithms. We introduce open format analysis data, and we screen for hundreds of thousands. We have now more than 100,000 substances in this platform. And then we have the semi-quantitation algorithms, and these algorithms produce semi-quantitative results or quantitative results. It depends on what we have in place in the laboratory. And then we can visualize the data in a map. From several points of Europe, we collect data because they registered and use the system. And then this is an infrastructure that also regulators are using now, that they can log in and retrieve for specific substances the data that we have in place. We have developed a series of algorithms to support this identification process. I won't go into detail with this. This is the semi-quantitation algorithms with the classical electrospray HRMS that we are using, and this is the new one with ion mobility data from these powerful instruments. Why they are different? Because these instruments have also very high sensitivity. And this is a figure that shows that, for instance, for PFAS, you know the problem with PFAS, are a range of substances that have unique properties, not only in the environment, but also in analytics. Therefore, we have developed an algorithm only for PFAS to screen and semi-quantify more than 4,700 PFAS that we are searching for in our samples. And of course, there was also a unique workflow with these very sensitive techniques for GC amenable substances. In any case, we have also workflows for non-targeted analysis. Non-targeted analysis means that we are prioritizing peaks out of our analytical methods, substances, based on the question we are posing in the samples. For instance, which substances differentiate two different classes of substances, for instance. Or if we are searching for new substances that have chlorine or fluoride, we are using some algorithms to prioritize these peaks and then try to identify. In any case, I'm not going to go into details, just mentioning the range of analytical techniques and methodologies we are using. And I'm just switching. This is a prioritization tool for an example. This is an example how we are monitoring a river, for instance, and with algorithms, we are prioritizing peaks that have a specific pattern, that are appearing every week, for instance, or in a specific time pattern, or they're having increasing trends or decreasing trends. These are spills in a supposed rivers, for instance. And then, with non-targeted analysis, we prioritize, pick, and identify what is this. So there are many, many capabilities to reveal what is hidden and, of course, skips from the regulatory monitoring programs. This is the concept and the idea. And this is how we have identified the sources of pollution in the Black Sea from Danube and Dnieper with non-targeted analysis and annotate substances to sources. In any case, the interesting part of the talk for my perspective and also yours, I think, is what we have found from several monitoring campaigns that have been applied or are applied in the, food chain. Life Apex is a project finished that we applied these methodologies in predators and their prey in the food chain from European specimen banks and natural history museums, meaning that we have several prey fish, for instance, or other, from marine and inland terrestrial environment, from 20 countries, applying these methodologies, and the results showed that we have found several substances to be biomagnified that were not expected to be biomagnified. And these substances are pharmaceuticals and several metabolites of pharmaceuticals, of course, plant protection products, biocides, surfactants, PFAS, okay, and several other substances like coffee-related substances or nicotine-related substances. And you see here that the concentrations of those substances in the predators are always higher than their prey, and for all this range of substances, of classes. And if we go back to the registration files of these substances, we don't find any property that shows that these substances should be biomagnified. Why they are biomagnified in the food chain? Because they are pseudo-persistent. They are released continuously in the environment, either in high or low concentrations, but they are persistent. So, biota organisms are accumulating these substances, okay? And of course, PFAS is a known problem. You know that PFAS have biomagnification properties. Of course, there are higher concentrations in predators than their prey. And we had the chance to have a time trend analysis 20 years ago, and we have seen the decrease from the ban of PFOS and PFOA in predators, but 10 years ago, the baseline concentrations remain stable. Thus, this means that these substances, because they are everywhere, they have a constant background concentration at the moment everywhere, even in our bodies, okay? So PFAS are here and will remain even if they are banned. Now, in the North Atlantic region, we had another project with OSPAR, and we have seen similar results in a range of marine organisms. Of course, all of these are food-related organisms, not only fish, but mussels, shrimps, whatever. And we have seen also concentrations, high concentrations of substances that we couldn't expect to be so. Like, for instance, psychoactive substances, tramadol and their metabolites, for instance, or venlafaxine and their metabolites. There are several pharmaceuticals and plant protection products that are biomagnified in the food chain, and they're, of course, through food chain are coming to back to us. And the last effort we have now is only in the terrestrial environment. We have several case studies that we try to screen, to analyze from soil to plants to insects to rodents to predators, to have in several countries all over Europe the same information in order to push for a better chemical management and better registration of chemicals from the chemical industry, because it is obvious that chemical hazards are there, here, everywhere, and we are exposed continuously, and through food chain as well. Therefore, food-related samples are very important. This is why they are included also in PARC, this partnership huge project that is running now. And just two more slides to show that these results, especially from the aqueous environment, were then studied for the risk in 5 trophic levels here, with very powerful QSAR models we have developed with the cooperation of UBA, the Federal Agency for Environmental Protection of Germany, from algae to rats. But now, with their support and some resources from the States, from Yale and FDA and EPA, we have developed Tox AI, which is an open source application platform to perform in silico risk characterization and environmental fate analysis with data for more than 100 endpoints, 105 from algae to mammals. Not only we have the endpoints, predicted endpoints from the analysis we are doing, but we have also predicted properties for the identified substances related to the fate, the environmental fate of the substance, and we have included in the platform also the biotransformation of these substances from bacterial, microbial, mammalian, whatever metabolism. And all this information, of course, should come with the uncertainty that should accompany any result for regulatory purposes. We are at this specific point to investigate what is going on in terms of chemical hazards assessment in the environment, and of course under One Health concept, which means that we have one problem. Either we are talking about biodiversity or we are talking about humans. We are one, and then everything all of these are related to everyday work we are doing. Thank you very much for the attention. I would like to thank also all the people that have worked for this project.